హలో ఫ్రెండ్స్, వెల్కమ్ టు ఫార్మా సిండ్రోమ్ యూట్యూబ్ ఛానల్. సో ఈ వీడియోలో మనం డిఫరెంట్ స్టెయినింగ్ మెథడ్స్, ఆ స్టెయినింగ్ మెథడ్స్ ఏ విధంగా బ్యాక్టీరియాని చంపేస్తాయి, ఆ స్టెయినింగ్ మెథడ్స్ ఎటువంటి ఇన్స్ట్రుమెంట్స్ ని స్టెరిలైజేషన్ చేయడానికి యూజ్ చేస్తారు. సో ముందుగా ఈ వీడియోని చూస్తున్నందుకు థ్యాంక్ యూ. సో మన యూట్యూబ్ ఛానల్ సబ్స్క్రైబ్ చేయండి, ఫార్మా సిండ్రోమ్ యూట్యూబ్ ఛానల్. సో ఫార్మసీకి సంబంధించి థియరీ, కాంపిటేటివ్ ఓరియంటేషన్, అలాగే ఫార్మసీకి సంబంధించిన జాబ్ అలర్ట్స్ గురించి అవేర్నెస్ ఇవ్వడానికి ఈ ఛానల్ మీకు హెల్ప్ అవుతుంది. అండ్ డెఫినెట్ గా మీరు థియరీ ఓరియంటేషన్ లో కావచ్చు, కాంపిటేటివ్ ఓరియంటేషన్ లో ప్రిపేర్ అవుతున్నా కూడా డెఫినెట్ గా మన ఛానల్ మీకు కనెక్ట్ అనేది యూజ్ అవుతుంది. సో సబ్స్క్రైబ్ టు ఫార్మా సిండ్రోమ్ యూట్యూబ్ ఛానల్. అండ్ గోయింగ్ టు ద ఇన్ దిస్ సెషన్, సో we will discuss regarding various method of sterilization, its principle and application. So first one is the sterilization method. Next, the mechanism by which the method will shows action, and application, which objectives can be sterilized by particular method. Sterilized by particular method. So first one is a dry heat. First method is a dry heat method. So dry heat nothing but a hot air oven 180 degrees centigrade for 2 hours or 45 minutes at 260 degrees centigrade. So here the mechanism how bacterium can be killed by oxidation of proteins in the bacterium. And this dry heat method, hot air oven method for sterilization of glassware, scissors, scalpels, ointment tubes, oils, powders in small container, and oily injections also. And next, moist heat, nothing but autoclave, 115 to 117 degrees centigrade for 30 minutes. Here, by using autoclave, in the autoclave, how the bacteria will be affected by the denaturation and coagulation of proteins. Proteins get inactivated, proteins get dysfunctional of the bacteria. So in this autoclave method, the substance like intravenous fluids, rubber and plastic items such as nylon as well as PVC bags can be sterilized by using autoclave. And next one is the tyndallization. It is a fractional sterilization, heating the medium 80 degrees centigrade 1 hour on each of 3 successive days. So tyndallization is a fractional sterilization, heating the medium at 80 degrees centigrade for 1 hour on each of 3 successive days. And this sterilization is for sterilization of objectives like surgical dressings as well as culture media. Next, pasteurization holder method. In pasteurization holder method, 62.8 degrees centigrade for 30 minutes, and flash method for 71 to 70 degrees centigrade for 15 seconds. So this is very very important. Many times they ask the questions, sir. So the holder method in the pasteurization, 62.8 degrees centigrade for 30 minutes, and in the flash method of pasteurization, 71 to 70 degrees centigrade for the 15 seconds. And especially, do you know, by using pasteurization, milk products will be sterilized. So this is simply regarding hot air oven, autoclave, sterilization, pasteurization. So what is the mechanism? Which mechanism they sterilizing the objectives? Okay. Next, some of the gaseous methods also using for the sterilization. In that, first one is using ethylene oxide. So, all the mechanism how ethylene oxide affect the microorganism. So, it causes what it is causing? It causing alkylation of sulfhydryl imino carboxyl hydroxyl groups of the proteins. So alkylation. So even if you remember anti-cancer drugs, so in the anti-cancer drugs there is a alkylating agents like many number of drugs are there. So that alkylating agents, what they will do? The alkylating agents causes alkylation of the DNA of the cancer cell. So alkylation of DNA inactivate the DNA of cancer cells, decrease the further cancer cell growth or cancer cell division. And by using this gas, substance like especially thermolabile materials, plastics as well as rubber can be sterilized. And next one is the formaldehyde. So formaldehyde is one of the gas. It is also acting as a alkylating agent and especially for the fumigation purpose. For the fumigation purpose. I hope you are from the B Farm. Definitely you also know that in your lab there is a performing fumigation by closing the sterilization area. Fumigation by using formaldehyde. Next, beta propiolactone. Beta propiolactone. Sterilization of the operation theaters as well as aseptic rooms. Sterilization of operation theaters as well as aseptic rooms. And next, one of the important one is HEPA filter test. HEPA filter test. Regarding HEPA filter efficiency test, so check efficiency of HEPA for high efficiency particulate air. HEPA means high efficiency particulate air. For air sterilization, dioctyl phthalate or DOP smoke test is performed. So HEPA filter remove particles larger than zero point 0.3 microns. So HEPA filter can remove the particles having zero point 0.3 micron size. 0.3 micron size. Okay. So here in this regard also there is a one of the point I have to discuss here regarding the phenol coefficient test. Regarding the phenol coefficient test. So especially regarding the phenol coefficient test, the common MCQ they are asking. So generally in the phenol coefficient test, the testing organism is a Salmonella type B. In the phenol coefficient test, the testing organism will be the Salmonella type. So many times they ask for the question. Many times they ask for the question. So what is this? So it is considered the disinfectant standard against which all other disinfectants are combined. So disinfectant, the standardization of disinfectant by using phenol coefficient. In the phenol coefficient, the testing organism will be the Salmonella type. The testing organism will be the Salmonella type. And this is also one of the tests for standardization of the disinfectant. Means how much disinfectant will be the effective. So that method is Kelsey Sykes test. A bacterial inoculum was added to the disinfectant in three successive labs at 0, 1, and 5 minutes. So testing organism for this test is Staphylococcus aureus, Escherichia coli, Pseudomonas aeruginosa, Proteus vulgaris. Proteus vulgaris means so these organisms using in test means how disinfectant killing this organism. So how effectively the disinfectant killing the organism. If disinfectant pass the test, that is an effective disinfectant. So for the standardization of disinfectants, so generally we will use disinfectant for the different purpose for cleaning, for cleaning water. So the disinfectant standardization by using different methods, one is Kelsey Sykes test and one is a phenol coefficient test. In the phenol coefficient test, the testing organism is Salmonella type, and in this test, the testing organism is Staphylococcus aureus, E. coli, Pseudomonas aeruginosa, and Proteus vulgaris. Proteus vulgaris. Okay, so this is today our session on the important topic of pharmacology for the pharmacist preparation. So like these videos, so subscribe to the Pharma Syndrome YouTube channel, and definitely you will get useful information and share with your friends also. Thank you.